So first of all, thanks very much for the invitation to speak at this conference. It's a great opportunity, and apologies for the little hiccup there as we started, but hopefully it goes smoothly from here. And thanks also for the kind introduction. So today what I'm going to talk about is considerations around the acquisition and artifact correction in multiparameter mapping, a particular protocol that's increasingly being used for quantitative MRI. So first of all, we have to ask the question of what is it that we're trying to achieve? And really, if we look at the brain microstructure, it's very complex. If we look within the grey matter, then we can see that there's a very rich cyto and myelo architecture that varies across layers and across regions of the brain. And it is information like this that we would like to be able to try to access using MRI, which is ideally suited to doing so because of its non-invasive nature. If we look within white matter, there's also a very diverse microstructure with different cell types, fiber pathways connecting different regions. And again, it is this type of information and how it underpins our everyday behaviors that we would like to be able to access. And indeed, to understand how changes in the organization of the brain microstructure underpin neurodegeneration or any other type of clinical condition. So the way that we typically use MRI is to acquire images that are weighted to particular physical parameters of the tissue. So for example, T1 weighted, PD weighted, or T2 weighted images. But what we can also do is, with our knowledge of how the signal has been created through physical models, we can actually compute specific physical properties of the tissue. So for example, to characterize their relaxation, or to quantify how much water content there is in a given voxel, or to characterize the diffusion properties of the tissue itself. And the reason that we can do this is that these properties of the tissue are sensitive to that underlying microstructure. The values and the rates of relaxation are going to be dictated by what kind of iron content there is, what kind of myelin distribution, how the fibers are organized, how the molecules can rotate, move, have protons exchange between different environments. And so they've got a rich sensitivity to that underlying tissue microstructure. Now, really what we would want to do is go a step further and use biophysical models that tell us how these relaxation times are being dictated by the underlying biological features, so as to be able to map things like myelo architecture, iron distributions, really quantify what the fiber pathways are like. But for today, what I'm going to do is focus on this middle aspect of quantitative MRI, because it is something that's become much more mature, but is also then a requirement in order to move towards this concept of in vivo histology, where we really map the underlying tissue properties. So as I mentioned at the start, the protocol that I'm going to focus on is multiparameter mapping, or MPM. And what we do in this case is acquire data with a very simple acquisition. It's a gradient echo. We can acquire multiple different images at different echo times. And in doing so, we can look at how the signal we've created relaxes over time or decays away. And this tells us again about that underlying microstructure, because there could be factors there that facilitate the more rapid relaxation. For example, if there's iron accumulation in a particular area, or indeed in the vicinities of fibers that are richly myelinated and highly organized. What we can also do is change the flip angle of our acquisition, and now we have a variable flip angle approach to mapping the longitudinal relaxation rate, R1, which again tells us about a different aspect of the tissue microstructure. It tells us about how the signal is recovering after it has been disturbed. We also have the flexibility to add in a pre-pulse. This could allow us to target specifically macromolecular or bound pool content, so things associated with aspects of the tissue like myelin or other cellular markers, and be able to then get a sensitivity to that. So in this case, you see that because of the rich myelination within white matter, the signal is decreased by applying this pre-pulse, and then looking at the magnetization transfer effects that occur subsequently. And again, we can quantify this. We often need to acquire calibration data also, so that we can correct for imperfections in our system, because ultimately we want to be able to compare these measures and have them truly quantitative. But if our system isn't working in the perfect way, for example, we've got errors in our transmit field or in our receive field, we've got variability. We need to take account of this. And I'm going to go into that in a bit more detail. But then ultimately what we can get by combining these types of data is multiple different metrics that each have a differential sensitivity to the underlying biology. So if I highlight some regions, you can see that as we move across the different quantitative metrics, the appearance of the tissue is changing quite substantially, and the contrast between different structures is also changing. And having this multimodal view is very beneficial because although we don't have a one-to-one mapping to the underlying tissue biology, we can look for what kind of biological changes would be consistently manifesting across the different quantitative metrics that we've measured and that we see differences in. Another important aspect is the fact that these measures are quantitative. So this means they're standardized and they have particular units. For example, for the relaxation measures, this is in per second. And that means that if I measure that quantity today and tomorrow and a week later or a year later, I should be able to compare those values and therefore be able to track longitudinal change. I should also be able to compare between groups, for example, looking at patients and how they differ from a healthy control cohort, or indeed to be able to pool patients from different sites because of that standardized nature. But this is a difficult thing to do, and so we need to take great care about it. This type of protocol is very efficient, and we can get sub-millimeter resolution in total protocol times of less than 30 minutes, and that can be with whole brain coverage. So this is what makes it very appealing. But as I said, it needs to be very robust in order to have sensitivity to longitudinal change or to inter-individual differences. And to achieve this, we need to consider multiple factors. So for example, what would be the impact of motion within or between acquisitions? We often think of it in the within case, but not so often in the between case. We need to consider what field inhomogeneities might exist, so the imperfections in our system that I mentioned before, and how these could lead to errors and need to be accounted for. It's also very important to consider what model assumptions are being made when we apply our models so as to extract the quantitative metrics. Are these assumptions valid? Under what conditions are they valid? So the next question then would be, how can we make it robust? And this is particularly important if we want to deploy this type of approach for use with clinical cohorts. So one aspect that I want to discuss is interscan motion. If we consider a case where we have two different images that have been acquired in separate scans and that are each of very good quality, we can think that motion is therefore not causing us a problem. But actually, if there has been motion in between, and this can happen, for example, if someone wants to resettle themselves, if they're having rather a long scan time, it can be nice to just take a breath, scratch your nose, but movement like that can lead to subtle changes in position. And if you think about the fact that the person is lying within a head coil that has a differential sensitivity across space, then any small movements can lead to a differential modulation of the images. And we don't tend to take that into account with our modeling. We assume that it's fixed. We do tend to account for the fact that there might have been motion between our acquisitions, and so we could do a rigid body realignment. But here what I'm trying to show you is that the modulation from the coil sensitivity may still be different. And in this case, if we compute, for example, an R1 map in this case, which does require two different acquisitions as a minimum, then we can have substantial errors. But actually what we can do is get some very simple calibration data in order to account for any changes in the receive field modulation that might have occurred as a result of motion. And then we can correct for that and restore the validity of our model and therefore the accuracy of our estimates. Really what we want to have is that if we have the receive field isolated from two different acquisitions, that it be identical. But if we look at the relative receive field, then I show you the distribution of what this value is across space. And we want this to be 1, but actually you can see that it could be as much as plus or minus 20% of that value. And this can lead to very substantial errors. If we look at the transmit field, so the efficiency of our excitation and how well that has been achieved, we see that although it might be inhomogeneous, it's relatively independent of position. So now we get a much sharper peak around 1. But if we move to higher field strengths, so for example, using 7 Tesla MRI, then what we see is that there's a much broader distribution of those values, meaning that the transmit field itself is also becoming very sensitive to our position. And this again is going to be another source of error, even though the images themselves might look quite okay. This is a little bit busy, but what I want to do is just illustrate the point that these different factors are contributing. So on the top row, we see R1 maps that have been computed in different ways. And in the bottom row, we see the error relative to a reference. So if we focus on this one that's highlighted by the orange box, this is the case of where there has been intrascan motion. And so in the R1 map, you can see that it's got a rather non-uniform appearance. The values are not being dictated solely by the biology. And at the bottom, that corresponds to regions where we've got very bright red or blue. If we account for transmit field changes, we can reduce that error to some extent. But the much bigger source of error, even at 7T, remains the receive field. And so if we account for that, which can, as I say, can be done very simply, then we can greatly reduce the amount of error that we have. But I think what's important to notice is that there is actually 2 sources, both the transmit and the receive field. And so our most accurate measures come by accounting for both. But this isn't so easy to do because to get very accurate transmit field measurements takes quite some time. So we start to increase our protocol duration if we were to do this. But the receive field can be done in a matter of seconds and therefore is something that should be integrated into practice. Now let's consider a different way in which we might introduce errors into our measurement. And that's by having assumptions that may or may not be valid. And what I want to focus on specifically is the multi-compartment nature of the tissue. So we saw at the start that actually within grey and white matter, there's a very complex microstructure. And so there is a lot of heterogeneity within a voxel. And we don't always account for that. So let's take, for example, an estimate of R2 star. So this is telling us how the signal is decaying over time. So as we sample across different echo times. These maps are very useful because they can tell us about distributions of iron content, for example. So you can see the basal ganglia are appearing with high R2 star values because of their rich iron content. And also myelination and higher myelin levels, as you see in particular fiber pathways, such as the zoomed in region of the corpus callosum, can also lead to more rapid decay. But here we've obtained this R2 star map with a flip angle of 6 degrees. Now we wouldn't ordinarily assume that the flip angle was going to impact our measurement. And in fact, we typically assume that it does not. But here, if I show you different R2 star maps that have been computed as the flip angle has changed, you see that this isn't the case. And in particular in the corpus callosum, you can see that the R2 star is changing quite substantially. And this is because we're actually assuming that we have a single compartment when we fit our R2 star, typically. And if this is not the case, then the apparent R2 star that we fit in that case will depend on the flip angle because we're going to have differential contributions from different subvoxel compartments of the tissue microstructure. We've recently shown that you can derive a metric that is independent of flip angle and therefore is more robust, and something that could be integrated into protocols like multiparameter mapping, where you do have multiple flip angles, which is required to factor out this effect. So what's causing it? Well, in reality, the signal decay is going to be affected by these subvoxel compartments. And if we think of white matter and think of a myelinated fiber, then what we have is water that's sitting within the myelin sheath itself, and also water that's sitting within the axon or outside the axon. But these have very different microenvironments, and so they have very different relaxation behavior. And so as we change flip angle, our sensitivity to these different compartments changes quite substantially. So here again, I show you 2 different R2 star maps that have been computed with different flip angles and highlight regions where the R2 star is changing quite substantially. Now, because we have this sensitivity, we can turn that into an advantage as well as considering it to be an artifact, and we can actually seek to compute what is the myelin water fraction. And this is very appealing because it's something that we could do retrospectively to existing data. or simply add to our repertoire in the multi-parameter mapping context. So now, let's take the case that we actually have robust data. Why might it be useful? Well, we could use R1 maps, for example, as an in vivo myelin marker. And here we see an example of where the R1 maps have been inflated onto a cortical surface. And the highest R1 values are corresponding to regions that have high myelination and that have been identified previously in studies of myelogenesis. And here we see a schematic from work that was done over 100 years ago now, looking at where these highly myelinated regions first start to appear over the course of life. And you can see that there's a very good correspondence between these schematics and the measures that we're getting in vivo. But the really important thing is that a lot of this information was being obtained ex vivo. And so having such an in vivo marker is really important because it means that we can start to look at structure in conjunction with function, which obviously isn't possible post-mortem. So here's an example of mapping the visual response to the retinotopic response to a visual presentation so that we can map out various different regions of visual cortex like V1. We could take the functionally defined boundaries from this type of retinotopic experiment and overlay those on R1 maps. And now what we see is that there's a very nice correspondence between functional and structural boundaries, indicating the functional relevance of this type of a measure. We can also look at how the brain might age over time. And this is important so that we can understand what normal aging might look like, so that we would be able to differentiate that from cases of neurodegeneration. And I think you'll hear much more about that in the next talk. But here what I would want to highlight is the fact that we can see this very widespread pattern of aging, and we can see that by using these quantitative metrics. And we can also then interpret what that might mean. So instead of looking at the metrics themselves, we could interpret that the green regions are corresponding to areas where there's an iron accumulation over the lifespan. And we could look at the red regions and interpret those as areas where demyelination is occurring over the lifespan. But again, we're getting this distribution now across the entire brain, something that is not easy to do post-mortem, where we typically focus on very small samples and specific regions. Thinking about neurodegeneration, we have a very exciting opportunity, particularly with the advent of ultra-high field, of being able to look at selective degeneration across different lamina. 7T offers this possibility because of its higher sensitivity and therefore our capacity to get much smaller voxel sizes. And what we would expect is that there would be differential regional and laminar profiles depending on the particular type of neurodegeneration that we're looking at. And we may be able to see much earlier markers than the more end-stage type of measures that are more commonly used at the moment. And an example would be predicting a reduced relaxation rate and R2 star measure, longitudinal relaxation rate and R2 star measure that would reflect neuronal and myelin loss in deep but not superficial layers in Huntington's disease. And this is an example of a type of hypothesis that's being tested at the moment within our centre. I also just wanted to finish off by letting people who are interested in taking up these methods know that there's some help out there to get started. So we have, as part of a consortium, developed a toolbox that tries to make it a little bit easier to analyze these types of data, and that integrates the types of correction schemes that I've been talking about, and many others. There's some documentation provided on our website so that you can get started on installing, using the data, using the methods, I should say. And then also we provide some data so that you can actually go ahead and compute some maps for yourself and make sure that you understand what the different correction schemes are doing and what would be the right way to analyze the data. So with that, I just wanted to finish by thanking my group, many of whom are pictured here, and thank you all for listening.
Thank you so much for this great talk. I'm just going to ask Olivier if there is any question in the chat. Otherwise, I have one.
Yeah, there's one question is in the chat from Dr. Mustafa Bourara. So he says, fantastic work, Dr. Callaghan. I was wondering what distinguishes MPM from MR fingerprinting. What would be the advantages and limitations of either technique?
Very good question, and a very interesting one. I think fundamentally the difference would be that in this type of approach, we have very well-defined signal models, and then we effectively perturb one parameter at a time. For example, we would change the echo time and acquire multiple images to quantify R2 star, or we might change the flip angle so that we could quantify R1. But we're doing that in a kind of a one-dimensional way, and assuming that everything else stays static. Whereas in MR fingerprinting, the approach is very different. And over the course of the acquisition, things like TR, flip angle will all be changing. And then you would look to see how did that signal evolve over time, match it to a dictionary, and then determine what would have been the consistent R1 and R2 star that would have manifested that particular signal type. So they're fundamentally very different approaches, and each will have their own merits and limitations. But I think what's actually rather common about them is that it's going to boil down to how well you can model your signal. Because fundamentally, that's how you populate your dictionary to be able to estimate what parameters would have given rise to this very complicated signal over time. Whereas in the more classic case of varying one particular parameter at a time, you also do that under the assumption that this is an appropriate signal model and that will allow you to recover the parameter that you're interested in. But in either case, there are factors that we do not consider, for example, the effect of flip angle on R2 star decays, as I was highlighting here, that we don't, I shouldn't say that we don't consider, but that we don't always consider. And how sensitive one is to those may depend on the approach and needs to be carefully looked at so that we can really understand exactly with a given approach what the benefits and limitations are.
Thank you. There's one more question in the chat. There's actually a couple more, but I'm not sure if we will have time for all of them. But there's one from Matthew Upman, who asks, is there any direct comparison between the iron maps and the myelin maps? So, for example, do regional changes in iron relate to regional changes in myelin, or vice versa?
So I think there the question is whether or not one could perhaps disentangle the various effects to know what was causing them. If it is that, I mean, I think there what's beneficial is this multimodal view. And being able to see what might plausibly be leading to the changes that are observed. But I think that's exactly one of the difficulties where there can be a many-to-one mapping such that it isn't always clear what might be the underlying biological change. So I think there needs to be a lot of validation around how one interprets. And again, having that more multimodal view is really helpful. I wanted to say something else to that direction. Yes, that ultimately, I think exactly having that understanding is what would motivate us to try and push further beyond qMRI so that we're not measuring something that does have multiple influences, but rather we're going back to the underlying tissue biology. And I think that's where the importance of the in vivo histology approaches really comes in, so that we can actually go back and say, this is really a change that is driven by myelination, even if it would manifest in a similar way, albeit not identical, but a similar way to how a change, for example, in iron content might.
Thank you. Maybe one more question. There's one from Nicholas Buck. So there are several methods to measure B1+. Do you see this method coalescing to a do you see this work coalescing to a best method that you would use or recommend people use?
I think it's a very important point, and there are really a lot of different methods, sometimes, you know, designed more for one purpose than another. I think a challenge for quantitative MRI is that it really has to be very accurate if we want to have high sensitivity, and that tends to mean more time-consuming methods. We've done some degree of comparison between methods to try and discern which might be the most suitable. But I think oftentimes it comes down to sort of common limitations that need to be overcome. For example, that you're mapping the transmit field, but actually what affects multiple methods would be B0 field and inhomogeneities there. So having, again, a sort of more complete model of what's going on where you can incorporate all of these effects might allow us to identify which would be the most appropriate method. I think oftentimes what people do in reality is to rely on methods they have experience of so that they can at least understand within context what the pros and cons are.
Okay, so I think we are going to move forward.